Hi, my name is Tsubasa Seto. I'll talk about my research. Noradrenaline activates ependymal ciliary movement through adrenergic beta-2 receptors. First, let me introduce background and purpose. Ependymal cilia are motile cilia that cover the surface of ventricles and provide cerebrospinal fluid called CSF fluid. CSF is secreted in the cold plexus of ventricles and circulates within the brain and spinal cord, during which it contributes to the maintenance of brain environment by nourishing neurons and removing waste products. It has been reported that CSF retention increases brain pressure, causing hydrocephalus, and increases the risk of decreased brain function, including dementia. Therefore, activating ciliary beating and increasing CSF fluidity are expected to prevent the deterioration in the brain function. In this study, the noradrenaline called NA and adrenergic beta-2 receptors called beta-2 agonist procaterol activate beating of ependymal cilia and investigated their signaling pathways. Next, I'll talk about the method. In Figure 2 shows transwell used for ependymal cell cultures. As the first step, cells were prepared from the whole brain of newborn mice and cultured in a flask containing a growth medium with 10% FPS for 7 days. And second step, after reaching confluent, the cells were sealed on transwell filters and cultured in the growth medium with 10% FPS for 3 days. And last step, the medium in the upper chamber was changed to an FPS-free differentiation medium and cells were cultured for at least 21 days. Next, I will show you the measurement of ciliary beat frequency called CBF. Figure 3 shows the observation of ciliary beating. The filter was cut out and set in the observation chamber, and solution was perfused from the ventricular side. The movement of cilia was observed with an inverted microscope connected to a high-speed camera, and the ciliary beat frequency called CBF was calculated as the number of waves in 1 second. Next, Table 1 shows the reagents used in this study and their concentrations. Concentrations of phenylephrine, clonidine, and dopamine, respectively alpha-1, alpha-2, beta-1 AR agonist, used in this study are much higher than their ED50 values. On the contrary, the concentration of procaterol is comparable with that used in previous study of bronchial cilia. Next, I'll talk about the last part of method. Figure 4 shows the distribution of CBF values in ciliary cells. The x-axis shows CBF values and y-axis shows number of cells. In this study, cells with their CBF values higher than 10 Hz were used, shown by the gray zone. These values are comparable with those observed in the in situ experiments using brain slices. Next, the result is shown. Figure 5 shows immunofluorescence staining of ciliary cells with anti-acetyltubulin and beta-2 antibody. This is seen in single cell. Beta-2 was stained in green, and acetyltubulin as a marker of cilia was stained in red, and the nuclei was stained in blue by DAPI. As a result, beta-2 was expressed in ependymal ciliary cells and co-localized with acetyltubulin. Next, Figure 6 shows the activation of CBF by procaterol. The x-axis shows the time, and y-axis shows CBF values. The rate of change in CBF value was observed after 5 minutes of perfusion of control solution to ciliary cells, followed by 15 minutes of perfusion with 10 nanomolar procaterol. The result shows that the CBF value was increased and attained a peak in 1 minute after addition of procaterol. This peak value was sustained during the perfusion of reagents such as procaterol. The CBF value was gradually decreased after removing procaterol. Hereafter, the CBF values were expressed as the CBF ratio between baseline, shown as blue circle, and shown as red circle, peak values. The CBF ratio was compared between the treatment with control solution and reagents. Next, I'll show you figure 7. Figure 7 shows the activation of CBF values by noradrenaline and procaterol. Then increase the CBF values in a concentration-dependent manner with ED50 values of 5 nanomolar, up to 31% for procaterol and up to 38% for noradrenaline. Next, I'll show you figure 8. I studied the effect of adrenergic receptor agonist on ciliary beating. The activation of ciliary beating was most seen when the beta-2 AR agonist, procaterol, and noradrenaline were used. These results led me to focus on my research, adrenergic beta-2 receptors. Next, I'll show you figure 12. Figure 12 shows that the beta-2 AR stimulated pathway for activation of CBF. Generally speaking, procaterol stimulated the beta-2 AR. Now then, I have the following hypothesis about cilia. Adrenergic beta-2 receptor agonist, procaterol, and noradrenaline binds to beta-2 AR. It activates adenate cyclase, which raises the intracellular cyclic AMP concentration and activates ciliary beating via pKa, and phosphate such as axonal dyne light chain. To confirm this pathway, the following experiment was conducted. Next, I'll show you figure 9. I studied the effect of propranolol, beta blocker, on ciliary beating. CBF values were compared with those of controlled solution perfusion and procaterol and propranolol and procaterol and propranolol in combination. As a result, shows that propranolol didn't inhibit the CBF baseline in the absence of procaterol. However, propranolol inhibits the ciliary beating and decreases the CBF value stimulated by procaterol. Next, I'll show you figure 10. I studied the effect of forskolin, an adenate cyclase activator, on ciliary beating. CBF values were compared between those of controlled solution, procaterol, and forskolin alone, and procaterol forskolin in combination. As a result, the addition of forskolin increased the CBF values by up to 27%. On the other hand, procaterol didn't further increase the CBF values after pre-treated with forskolin. These results suggest that activation of ciliary beating by procaterol is mediated by activation of adenate cyclase. Next, I'll show you figure 11. I studied the effect of H89 as a pKa inhibitor on ciliary beating. CBF values were compared with those of controlled solution, procaterol, and H89 alone, and procaterol H89 in combination. As a result, H89 didn't inhibit the CBF baseline in absence of procaterol. However, H89 prevents the procaterol-induced increase in CBF values. This results indicate that ciliary beating by procaterol is mediated by protein kinase A. Finally, in conclusion, the stimulation of beta-2 AR by procaterol or noradrenaline activates adenate cyclase, which in turn stimulates ciliary beating by activation of pKa. Thank you for listening.